Hello, my name is Dr. Julian Barwell. I'm a consultant in clinical genetics at the Leicester Royal Infirmary. In this, the third of three videos looking at the launch of the Genomic Medicine Service, we'll be talking about what to do when you receive a result on your patient. This may be a specific gene test from the test directory or the result of whole genome sequencing, either from the germline, from the blood, or from the tumour. We ask you first to check the patient's identity, and this is a particular issue in genetics when we may be dealing with more than one family member who may have very similar names. Secondly, we ask you to go back through the medical record to see if there's a wider family history of the condition or related disorders, or any particularly unusual features which would make you think the disease may well be Mendelian. That means a single gene trait passing through the family. The third thing to do is to look at the actual result and see if a specific gene change or a chromosome rearrangement has been identified. If nothing has been found, then ask yourself, is there a wider family history or any other subtle phenotypic changes, such as anatomical abnormalities, which would mean you're still convinced there's likely to be a genetic cause? And if this is the case, we still recommend that you refer the patient to genetics, even if you're unable to find a mutation or what we call now a pathogenic variant. If a variant is identified in the report, it may not necessarily be disease causing. We all have approximately 400 subtle changes in our proteins, which we call missense variants, which may not necessarily cause disease. It is possible that the gene change that you found in your patient may be one of these. In fact, when it comes to subtle missense variants, whether or not that particular variant is disease causing changes depending on the gene itself. There are a number of ways in which we can start to understand whether or not that variant is likely to be related to the condition. Firstly, has it been seen before in any other patients with the condition? Has it never been seen in a control population? If you have a number of effective individuals in the family, you may be able to track the gene change. There may be a functional test in the laboratory that you may be able to carry out to be able to understand more about the gene change. The precise chemical nature and position of the gene change may also be important. Finally, sometimes we can look at databases through evolution to see if that particular part of the protein, the amino acid, remains always the same, be it in humans, other mammals, but also through amphibia, fish, and single-celled organisms such as amoeba. By looking at this, we can try and work out how important that particular part of the protein is. There are computer programs which you can look for these particular types of gene changes and help develop a score as to whether they feel that that particular variant is causing a disease in the family. If we feel with over 95% certain that the gene change is disease causing, then we're likely to call it a class 4 variant. If we are 99% plus certain, then we consider it a class 5 variant. The class 4 and the class 5 variants mean that we'll be in a position to offer a predictive genetic test to other relatives. Sometimes we are less sure. If we are approximately 80% sure that the variant is disease causing, we call it a class 3 variant. This may provide information about the condition, but by and large we cannot use this to help predict the risk for the relatives. If the variant is felt very likely to be benign, it is called a class 1 variant, and somewhere in the middle is a class 2 variant. There are databases which can be used to look at these in more detail, and the classing of the variant itself is now being standardized throughout the country as part of an American college guideline system, the ACMG. We hope to be in a position now to be able to work with you as part of multidisciplinary teams to look at the variants that your patient has been effective with, and in the future be able to discuss this more with patients. This is going to be a major part of clinical genetics in the future, and please do not hesitate to contact the clinical genetics department if you have any particular concerns. We would rather that you make a referral when you are unsure than to try and reassure a family and then we find out that other relatives are affected. This is the end of the video. I'd like to take the opportunity to thank you, and please feel free to look at the links at the end of this video and look at our other recordings on genomic medicine and the consent conversation. Thank you very much. Goodbye for now.